Hello dear viewers, welcome to the video on nitrosamine risk assessment for drug substances. In this video we are going to see how the risk assessment is done for active pharmaceutical ingredients or drug substances, and how this risk assessment and the nitrosamine presence in the drug substance is going to impact onto the drug product. So in pharmaceuticals, nitrosamine risk assessment is very much important, and nitrosamine impurity limits are very stringent. Also, nitrosamines are the class of compounds which are known to be cohort of concern, and these have the carcinogenic and mutagenic potential. So, nitrosamine impurity assessment in the drug substances and also in the drug product is becoming very important as the days are going. Nitrosamine is not a new topic for the pharmaceuticals, but as the nitrosamine are being investigated and are being controlled, so nitrosamine is becoming a critical quality parameter for the pharmaceuticals. So let's start with the video, and we will see what are the key criteria or what are the key risk assessment points. So risk evaluation is done for the drug substances for the presence of nitrosetting agents, then presence of or risk of nitrosamine impurity precursors, and the presence of amine functional groups. See, there is a chance of nitrosamine formation if some prerequisites are available, like presence of nitrosetting agent, then presence of acidic condition, and presence of the amine group. As the drug substances are prepared by a number of manufacturing steps, also for making the drug substances, raw materials are required, and that's why the risk evaluation is very critical for drug substances. And three out of the all key points I have enlisted here is presence of nitrosetting agent, presence of amine impurity precursors, nitrosamine impurity precursors, and the amine functional groups. So if any presence is there, then there is high risk of nitrosamine formation. And risk evaluation is done throughout the API research, API commercial production, and the API stability. First of all, the risk assessment involves raw material risk. Whether there is any nitrites or nitrates present into the starting materials, intermediates, or the key starting materials. Whether there is any presence of nitrites or nitrates, whether there is any contamination of these nitrosetting agents. Nitrites and nitrates are the nitrosetting agents. After raw materials, there will be the use of solvents in the API manufacturing. So, use of the recovered solvents or contaminated solvents may have the additional risk for nitrosamine impurities. Also, use of contaminated reagents, catalyst may have an impact on risk. Recovered solvents are those solvents which are used in the manufacturing of some APIs, and these are recovered, and then again these are used for the processing. So there are high chances of contamination of these recovered solvent. Then is the amine group. So if amine group is a functional group in the APIs, then there are chances of the nitrosamine formations. If there is presence of secondary, tertiary or quaternary amine group, and if the ammonium salts are being used in the manufacturing process, then there is possibility of nitrosamine formation. The another most important thing is degradation. If there is any degradation of starting materials, intermediates, reagents, or the drug substance itself, that is API itself in the process, and generation of nitrosamines is there, then there is a high risk for the nitrosamine presence. Risk of contamination and cross contamination. Contamination is from raw materials, starting materials. Sometimes the starting material is called as key starting material, that is KSM, and the equipments used in the same manufacturing area. If a API plant is having a dedicated area for some API, so contamination may be there from raw materials, starting materials and the equipments. Also, cross contamination may be there from the manufacturing area which is away from the actual manufacturing area, and it is not the same. So that risk is also there. Cross contamination from equipments in the different area. So this may also happen that the different area equipments may lead to cross contamination. Some of the times some movable equipments are used in the manufacturing area, and these equipments may become the reason of cross contamination. Use of nitrogen gas and air. In the API manufacturing, nitrogen gas is used and the compressed air is also used for different reasons and for different purpose. So quality control test for nitrogen gas and the compressed air should be there. Contamination chances and risk assessment for the use of nitrogen gas and compressed air should be performed, and there should be a check for filtration of nitrogen and the compressed air. If the filtration is not there, if the quality of the nitrogen gas and air is not as per the requirements, then these things can lead to the contamination for nitrosamine impurities. So these are considered in the risk assessment. Important thing is water quality. Water is used for cleaning. Water is also used as a solvent. So quality and quality control test for water become very critical. Water is used in the plant which is purified by reverse osmosis, that is RO water, and also by the filtration technology. Sometimes the water is tested for presence of nitrates and nitrites, and if that water pass the nitrite or nitrates test, then only it is used in the manufacturing. So always have a check and risk assessment for the water quality. Then is the cleaning. So wherever batches are taken, after some batches cleaning is performed. Also, when there is a product changeover, that means you are manufacturing a drug substance A, its manufacturing over, and then you are going to manufacture the drug substance B. So in between these two, cleaning is there, that is called as product changeover. And if the batch of A is completed and again batch of drug substance A is getting started, that is called as batch to batch cleaning. So this cleaning efficiency have a great impact onto the contamination, cross contamination, and also from one product to another batch of the same product, or from one product to the another batch of different product. So product changeover and cleaning should be very efficient. The cleaning process, whenever is performed, that time the cleaning time, cleaning reagents which are used, these are required to be strictly followed, and there should not be any deviation for the cleaning. Packaging may also lead to introduction of the nitrosamines. Risk from the primary packaging materials, risk from the printing inks and the packaging processes should be taken into consideration. The primary packaging materials are those materials which are in direct contact with the drug substance. The printing ink which is used to print the batch numbers and name of the API and the batch details like manufacturing date, testing, retest date, expiry date, etc. So it is known that some of the times the printing inks and packaging process which involve the heat has generated the nitrosamines. So packaging is taken into consideration for risk assessment. Then if the risk assessment is done and there will be two outcomes. First outcome will be that there is no risk for the nitrosamines, and another will be there is a risk for nitrosamines. Because in API manufacturing, lot of the synthesis steps are involved, lot of reagent, catalyst, solvents are used, lot of equipment and processes are used. So if there is a risk for nitrosamines presence, then listing and testing of the possible nitrosamines and the NDSRI should be performed. Also, any remote risk or possibility should also be taken into consideration. For example, if the API manufacturer is not adding any nitrosating agent, or it does not contain any amine group, or cleaning process is very efficient, nitrogen, air, water, or the raw materials are strictly controlled, but still if any of the raw material or intermediate is processed with some nitrosating agent, so that traces impurities may be brought by that intermediate. So any remote risk and possibility should be assessed. Then if there is a presence of nitrosamine impurity, then testing and control of specific impurities should be performed, and the stringent limit should be set. Any impurity in API which may increase the possibility of nitrosamine formation in the drug product needs to have stringent limits. In simple meaning, if you found that impurity A, for example, I am saying impurity A may increase the chance of NDSRI formation or nitrosamine formation in the drug product, so that impurity is acting as a precursor, or that impurity is being responsible for nitrosamine formation in the drug product. That's why that impurity is required to be strictly controlled, though it is below ICH limits, or though its risk is very low, but still the limit should be there. Whatever the limits are set, so that impurity should be routinely checked for its quantitative presence below 10% of the limit, or is it between 10 to 30% of the limit, or it is above 30% of the limit, so that impurity can be strictly monitored, and if required the process can be optimized and evaluated to avoid the higher percentage or higher quantity of that impurity in the API. Many of the times in the manufacturing of API, it is found that the impurity, though it is below some level, still it is responsible for the nitrosamine formation. And once you have the data of those impurities, then have the complete trend data for the drug substance batches, data of batches manufactured with different routes, data of commercial scale batches, and the stability data. All these should be evaluated critically and control should be there. Also include the specific impurity test and specification in the finished product. So that is the finished product part that required to be controlled. So this video enlists the possible risk points or key risk point for the nitrosamine risk assessment for the drug substances. I hope I have covered maximum of the points, but still the list is increasing. So always have update knowledge onto the nitrosamine risk assessment for drug substances and the drug products. Let's meet you soon with such kind of informative video. Till then, take care and bye bye. Thank you.